Stromatolites are rock-like sedimentary growth structures, formed by the trapping, binding and precipitating activities resulting from microbial biofilms in shallow waters. In 2017, scientists identified fossilized stromatolites in Greenland that date to about 3.7 billion years ago. In 2013, the microbially induced sedimentary structures, or myths, of stromatolites was found to be unchanged through time, up to 3.48 billion years ago. As stated in the article, Noffke has identified 17 main groups of such Miss-S textures caused by present-day microbial mats, and has found corresponding structures in geological formations dating back through the ages. Stromatolites host a diverse group of microbial life which include cyanobacteria, other photosynthetic bacteria, anaerobic heterotrophs including fermenters, sulfate reducers and sulfur-oxidizers, all of which are tied together by a network of syntrophic relationships. Syntrophy is the cooperative interaction between at least two microbial species to degrade a single substrate. This type of biological interaction typically involves the transfer of one or more metabolic intermediates between two or more metabolically diverse microbial species living in close proximity to each other. Thus, syntrophy can be considered an obligatory interdependency and a mutualistic metabolism between different microbial species, wherein the growth of one partner depends on the nutrients, growth factors, or substrates provided by the other else. This syntrophic interdependence is crucial, as the survival of one bacteria group depends on the existence of others. This essential syntrophy between bacteria can be seen in the 2018 article titled Microbial Mat Ecosystems, Structure Types, Functional Diversity, and Biotechnological Application. In short, if even one major type of bacteria group did not exist in these ancient and diverse microbial communities of stromatolites, then all of the other interdependent bacteria would soon die out. To state the obvious, this essential interdependence of the most primitive different types of bacteria that we have evidence of on ancient Earth makes the origin of life problem for Darwinists that much worse. Moreover, not only do microbially induced sedimentary structures, myths of stromatolites, show an extreme conservation of morphology, but individual bacteria do as well. As the article titled Static Evolution, Is Pond Scum the Same Now as Billions of Years Ago, found that ancient bacteria looked exactly like modern species, and the similarity in morphology is widespread among fossils of varying times. And fossilized bacteria show an extreme conservation of morphology, more extreme than in other organisms. This extreme conservation is found at the molecular level as well. In an article titled The Paradox of the Ancient Bacterium Which Contains Modern Protein-Coding Genes, it was stated, almost without exception, bacteria isolated from ancient material have proven to closely resemble modern bacteria at both morphological and molecular levels. This extreme conservation for bacteria at both the morphological and molecular levels is very unexpected for Darwinists, since, as Alan H. Linton, Emeritus Professor of Bacteriology, stated in his article Scant Search for a Maker, bacteria, the simplest form of independent life, are ideal for this kind of study, with generation times of 20 to 30 minutes and populations achieved after 18 hours. But throughout 150 years of the science of bacteriology, there is no evidence that one species of bacteria has changed into another. In conclusion, in the place we most expect to find evidence for Darwinian evolution, namely bacteria, Darwinists simply lack any evidence to support their claim that Darwinism is true. In fact, the extreme conservation of bacteria at both the morphological and molecular levels directly contradicts their claim that Darwinian evolution is true.